Thank you for joining the PATRIC webinar on our new service, the Comprehensive Genome Analysis. This is a new thing we've launched that I'll go through. I'll give you some discussion of why we're doing it, but I also want to encourage you all. We'll go through the webinar. There will be time at the end to ask questions, but always if you have any questions with PATRIC and you need help, you can go up here into the help section and you can click on provide feedback, and this will open up something that comes directly to me and to other people at PATRIC to describe your problem. If you have problems with specific jobs, I'll show you how to do that later on in the webinar. So all of you who have been using PATRIC, if any of you have been using PATRIC, you know that we have a number of services. So if I click up here on the Services tab, you can see that we have Assembly, Annotation. This is the new service that we're going to discuss today. Similar Genome Finder. All of our services are here. And when we started with giving these different computational pipelines to the people who use PATRIC, we generally try to choose things that biological researchers would have trouble with. Like doing an assembly is often pretty difficult. If you're doing something with spades, you have to download the pipeline, you have to learn to run it, you have to learn how to run it on your reads and interpret the results. So we are trying to make life easier for our users. The Comprehensive Genome Analysis is actually a combination of a number of different services in PATRIC that we are trying to give you a somewhat quick way to have an idea about the quality of your genome. And rather than how it used to be is you would do assembly, and then you would go in and do annotation once you did the assembly. And then you would go in and look to find the closest relative using our similar genome finder analysis. And then if you wanted to build a tree, you would go into the phylogenetic tree comparison. So these were all separate steps that we have in PATRIC that we have now combined into one service that we call a Comprehensive Genome Analysis Service. You can start with a read file, or you can start with assembled contigs. So you notice that the interface changes depending upon what you're starting with. So if I want to start with the read file, you can load paired reads, you can do a single read library, or you can use an SRA accession number. And then once you load those, you can go in and choose the parameters. And once you've filled everything out correctly, the submit button will turn blue and you will be able to submit your job. So we're going to step through this today. I'm going to show you how to submit a job. I'm going to show you how to track your job. I'm going to show you how to find the data once it's been completed and how you view it in PATRIC and interpret it from this particular analysis. So we're going to start with a read file, and you can follow along with me. If you have reads on your computer or you had an SRR accession number that you can put in, I have some reads loaded in the PATRIC database that you all can use step along with me. Now, anybody who's seen my webinars of late, if any of you have joined me for the other ones, you'll know that I have this obsession with Buchnera. Buchnera is an endosymbiont of aphids. It's they provide a similar function to the aphids that our mitochondria provide to us. And I've become really fascinated with them. They're a great bacterial genome to look at because they're very small and they assemble very quickly and they annotate very quickly. And also they're fascinating. So if you ever want to read up on Buchnera, I would encourage it, although I guess that sounds absolutely nerdy, but oh well, we're all on this webinar together, so I guess we're sort of in that category. The reads that I have available are single reads as opposed to a paired read library. Now you may want to know what types of sequences we allow on PATRIC. You can open the dialog box and it will give you information. Generally though, what PATRIC will take PacBio, Illumina, and nanopore reads. So let's find the file that we want to use. So to open it, and also to open a dialog with your computer, you're going to need to click on this folder icon that's right here. Because it's single reads, that's where I'm clicking. And so this shows the mess in my own private workspace. But I want to, if you wanted to upload something directly from your computer, you would choose the upload to current folder. And if I do that, it'll give me a dialog box. I would then click select file when this opens, and it gives me a dialog box with my computer. But actually I want to load stuff that I've already prepared for you in what we have in PATRIC, a public workspace. You can notice right here that I'm in my private workspace. This is my name and it tells me that I'm in my home directory. The reads that we've been using for workshops are in the public workspace. So if you click here on the down arrow and then on public workspaces, that'll rewrite the page and it'll show you all the data that we currently have in PATRIC that's public. And if we look down here, oh look, we have Buchnera reads here for the PATRIC workshop. I can click on this icon here, which I've been told is an icon of the globe, because I guess it means global and open to everyone. And clicking on that, it opens up in the public workspace, the Buchnera Reads PATRIC workshop. I can select that. Oh, that was fast. And well, I'm not sure why it did that like that. Let me try it again. Okay, there it is. And then I click OK. So you highlight it and then you click OK. Well. Let me put in here SR. Okay, here it is. For some reason, it was blocked from that particular place. Okay, and then we move it over here. This is an important part, is once you load the reads here, you transfer them over into the selected libraries. If you don't do that, this is what it's going to build the assembly on. Now, if we had paired in reads, I just want to point this out. Oftentimes, at least with our own sequencing facility, when I submitted Genome for sequences, they'll often run multiple lanes, and generally we do Illumina paired in reads. And the sequencing center will run multiple lanes with the same genome. You could enter each of those, each of the specific combinations, to make a better assembly. It'll give your assembly more power. So I've been told by the designers of the assembly pipeline that it's unlimited as to the number of paired in read files you have, although I'm assuming you don't have, that you didn't run unlimited lanes on the sequencer. But that's another way you can boost that. So here's our single read pair library. Here's the number here. So it's going to build an assembly based on this. So I can go in here, and then we've got to choose the parameters for the assembly. And these are actually a combination of the parameters that we use for both the assembly and the annotation pipelines in PATRIC.
So I got somebody's asking here, the Buchnera Reads folder is empty for me. I cannot see the SRS.
Okay. Someone has said that the Buchnera Reads folder is empty. Make sure that you're signed in, which you should be, to be able to see it. So you can see up here that I'm signed in. I hope you can see that. Let's go back. I'm going to delete this, and I'm going to show you another place that I have it, and I want you to try here. Just so you know, someone is working along with me, and they are having trouble finding the reads. Oh, you know what you can do? you could go into and put in here SRR, because that's where I got it, 4240359. So I'll leave it up there for a second so you can see it. And there, it's appeared there.
And that's where we're going to retrieve it from.
That's going to retrieve it from this sequence read archive. One more thing, because we've had this question. This is the easiest way for you to do it right now, but let me show you another place that these reads are. First, I want to cancel this and go back. I want to go into, want to go back to my parent folder. Oh, sorry. All right. And down here in the public workspace, we have a folder called, we should have a folder that everybody can see. That's not, the Keith was having trouble seeing it from there. But generally there's a thing called PATRIC Workshop. I may have to go into Home to see that, the global home. And then, well, shoot. I'm having a little bit of trouble seeing that right now. I'll work it out before the end of, we've got a simultaneous workshop going on in California, and it may be altering this. But for now, let's just use this and move it over. Okay, so PATRIC has a number of assembly strategies that you can use. When you choose the auto assembly, it runs 3 different assembly algorithms at the same time: Spades, Velvet, and IDBA. And it will run all 3, and then it'll choose the best assembly. We also have Fast, Full Spades, Kiki, MySeq, Plasmid, which is fast, which is Spades, sorry, and Smart, which is one we use for PacBio. Well, let's just use the auto assembly method right now. And so that's going to assemble those reads that it gets from SRR using the auto assembly method. Now we need to give some information, some things that we assume we know about the organism. So it's bacteria, better be that, or archaea, because that's all you can do in PATRIC. The one thing we need to do is give a taxonomic name for the organism. So I can start writing here, Buchnera. And actually this one, I know the species is Aphid E. coli. I don't even have to type all of it. As I'm typing, PATRIC is trying to find the closest match to this genus or to the name that you're putting in there. So you notice I click that and it gives the taxonomy ID. Now you may not want, you may not know what it is, or you may not necessarily want to make a taxonomic assignment. One of the reasons that it's good to get below the genus level is that PATRIC builds 2 types of protein families. One are the global families that cross the genus barrier, but we also do what we call as local families, which are genus-specific protein families. They're really excellent for comparative genomics. So I would encourage you to try to get to genus if you can, because I personally find those local families very informative, and I use them a lot. But you will have the global families. If you don't get to a genus level, it won't assign those protein families. It can't do it. Next, you need to provide a label that will stick on the end. Now, I want you to watch the name down here as I start typing. I'll call it CGA webinar. And so that tells me that this is the genome, and I always put something on it so I know when I did it. Today is the 8th, 08 OG. And it's important to, if you use the same name, PATRIC doesn't like it, and PATRIC will refuse to or it will give you an error. It'll give you a message that says you have to do a unique name for this. Now we need to do an output folder. What I'll do is show you how to create an output folder for those of you who don't know. You've just got to tell PATRIC where to put your data. So you notice if I drop this down box that I have a ton of different folders. And because I give a lot of workshops, I've actually annotated this Buchnera genome, I don't know, maybe 40 or 50 times, I can't remember. But I want to show you how to create a new folder. So to do that, click here and let me caution you, you're about to see my ugly workspace, and I apologize for the disarray that it's in. So this pops up. I can look here and I see, oh, I'm in my workspace. This is Rebecca Wadham's workspace. And I want to create a new folder to hold this data. And so you noticed I clicked on the folder icon here, not the upload icon, but the folder icon, and it allows me to create a new folder. I'm going to call it CGA webinar. And I'll create the folder. So the folder's created. And if I scroll down, there it is. So I can click on that, and hopefully it's going to show up there, the CGA webinar. Okay, so we've submitted the reads. We've chosen the assembly method. We've assigned a taxonomic name. We've given it a unique identifier that will be associated with the genome. Oh, I forgot to mention this. There are two different flavors of genetic code that we have at PATRIC. Almost everything we do for archaean bacteria is table 11 of the genetic code. However, if you work with Mycoplasma, Spiroplasma, or Uroplasma, you need to use genetic code 4, Buchnera's genetic code 11. And just the other day, I was looking at all, there are a ton of different genetic codes, but these are the only two we offer. Both both buttons are blue. Now I can click submit. And once I do, it tells me that the job has been submitted and it's going, or at least it's entered into the queue. Now let me briefly discuss some of the things that are the main problems that we have, that we get users asking about. The job enters a queue in PATRIC. The queue is first come, first serve. On a monthly basis, sometimes we get as many as 10,000 jobs submitted a month. Sometimes you're going to have to wait for your job to complete. This particular job, the comprehensive genome analysis job, if you've submitted it and it's still queued after a single day, please let us know and I'll go in and see if there's a stall on the service. We don't know that things are wrong unless people tell us. If the job starts running and it hasn't completed in a day, please let me know about that as well, and I'll show you exactly how to do that in a minute. I also, while we're here and on this page, I want to talk to you about the main problem that we have in running assembly jobs on PATRIC and where people go the most wrong. So I'm going to hit reset here just to wipe out the page. It happens when we're running paired read library files. The main thing that happens is that people pair the wrong reads together, and I'll show you how that can happen. Like, let's say I want to look at this guy, and I'm going to go in and take this and put this here, and this one is, let's say I chose that, okay? You can notice here, if I move them over, it's telling me that I can't use the same file. So that's a good thing. It's trying to give me something different here. But I need to go in and do read one. Read one for this. There. And then I can move it over. So this is the main place that where people get the most errors. Their assembly fails because they have not chosen the same mate pairs. And you can see even I had trouble with it, and I do this all the time because I've got the screen blown up a bit. You should always go in and check here on the information button to be sure that the reads match up and that you have an R1 and an R2 so that you have the forward and the reverse strand correctly paired together. Okay, so I've submitted a job. I'm not going to submit this job because I've run this one a number of times. You notice down here at the bottom of the page that you can see the status bar on uploads and on jobs. If I click on that, it'll show me my current jobs. And you can see here's this one, and it's currently queued. Now, you notice when I click on something that the green bar becomes highlighted with possibilities that I can do now that I've selected this. I can view the job, but there's nothing yet to view. I can kill the job. This happened to me just the other day. I submitted a job and then realized, oh no, I put in the wrong taxonomy. Oh, so I came in and quickly killed the job. You can, and this is the only time you can kill the job. Once the job is completed, it's on your jobs page forever. But this is the most important one. If this thing has been queued for over a day or it's been running for over a day, I need you to click on this icon that says report issue. I click on that and then I can say, my job has been queued for 24 hours. Give us some hint. Sometimes people don't put anything in here and I don't know what the problem is. So we need something for you to tell us what's wrong with this job. Often people will have a successful job and they'll report an issue and I don't know what the problem was. So I have to write them and say, it looks to me like your job is completed. What's wrong? But you can put that in there and then submit it. I'm just going to put it here because it's going to go to me and then I'll kill it. And so it's submitting that ticket and it'll send it. It seems to be a little slow right now. And there, it's sent off. And so if there's a problem, it comes to my email address and then I go into contact the developers and we try to see what's wrong with that job. So the workshop is more like a cooking show where we run it and then we try to show you the finished product. So you can see I've got one actually running that I started this morning, but it's still queued because there's currently a heavy load on the assembly pipeline. This assembly here is tied to this job. So when I run one of the comprehensive genome analysis jobs, I'll also see an assembly and an annotation for each of those in the job page. So let's look at a completed one. And as I look down here, you can see that I have been running a lot of jobs. And how am I supposed to find that? You can go up here and you can click on all services. And then I can go down to comprehensive genome analysis and click on that. And that's going to filter the table to show only those jobs that I've run recently. And here you can see a bunch of them are Buchnera. I've done a bunch of other things too with this. So this is a job that's been completed. So this is I call this the ATTC SRR genome. And you notice I can click here and then I can view that job. I can't kill it now because it's completed, but I can view it. So you need to click on the green eyeball here to see the job. And that's going to take you to the jobs page, or it's going to show you all the different reports that are associated with this job. It tells you the job ID number. If you've had a problem with this job, we need this number. This helps us track down what went wrong. It shows how long that job took to run, not how long it was queued, but once it entered into the assembly pipeline, It took 1 hour and 2 minutes and an unbelievable number of seconds, so disregard that. If I open the parameters, it'll tell me all sorts of things about that job, which probably you don't really care about, but it's there for the computer savvy people. Now, this is the data that we do care about. We prepared a document that shows all the information about your genome. And first, let's look at that, and then we'll go into these other files that are here. So I select that, and you notice that there are a number of things that I can do. I can move that file, I can copy it, I can rename it, I can delete it, I can view it. But because it's an HTML file, if I look at view it here, it's just going to show it in this small amount of space, and I want to experience it more fully. So I'm going to download that file just to my desktop, hopefully. Let's see, here are my downloads. Click on this. And here is the full report. And this is what is the beauty of this new service in PATRIC. Rather than you running a separate assembly, a separate annotation, trying to find the closest relative, running an initial tree to try to figure out who's closest to what, trying to figure out, looking at the annotation to see if there are any virulence factors or antimicrobial resistance genes associated with this genome. We've done this all for you here. Let me see if I can blow it up. So the report, the genome report, first starts and it tells you what the genome was, but most importantly, it judges. This is something that we've had users ask us for quite a while. I don't know if my genome's any good or not. Generally, I was having to do annotations and compare it to a close relative to determine if I thought that my assembly and annotation in PATRIC is of good quality. But we run a query that looks to do that, and it'll tell you if it thinks your genome is of good quality or poor quality, which would mean maybe you need to go back and do some more sequencing. Next, it tells you which algorithm. I had chosen the auto algorithm, but remember I told you that when I choose auto, it runs 3 and then it chooses the best one. And so of the 3, spades was the best. So it tells me that the genome was assembled with spades. And it also, we'll get to this in a second, it'll also give you the reference for that. Our goal with this is so that you can use this as the basis for submitting your genome to like genome announcements, which we all want to do if we have a kind of quality genome, so we get a publication with this. This is supposed to imitate that, and I want to provide you with the references so it makes it easy, very easy for you to do that. Along those lines, we try to provide the same statistics that something like genome announcements requires, so that you can take this information, put it in whatever the necessary format is for the genome, for the publication that you want to use or the journal that you want to use, and it's all there for you. It makes it very easy for you. So here, this part here generally is the information that's most important to you if you want to publish this genome. Next, it gives you a summary of the annotation. It, once again, lets you know which genome. It tells you what pipeline we use to annotate the genome, which we use RAS-TK, and down at the bottom you'll see the reference for the RAS-TK, for the reference for the publication of RAS-TK, and it also tells you the unique genome identifier. We also tell you what the taxonomy is. Okay, and it also, next, will give you, this is something else you would need for your genome announcement if you wanted, if you were going to publish this thing. Just this brief summary will tell you the coding sequences, the transfer RNAs, and the ribosomal RNAs, and if we had any other problems.
Rebecca, there's a couple of questions.
Okay.
How does PATRIC judge if a genome is quote unquote good? And what about depth coverage?
You know, we don't have the depth of coverage yet. That's something we're going to be adding into this. The question was, how do we determine if the genome coverage is, if the genome is good quality, and what do we do about depth of coverage? Depth of coverage, we're going to be adding this. We're working on adding this functionality now to our annotation service. To determine if the genome quality is good, it's looking at closest relatives and the lengths, the N50 and the L50 of the contig to determine, and those are standard things that you use to determine good quality, but we also look at similar genomes to see if it is within the same range. Like if you had, if this Buchnera genome instead, because we know generally that Buchnera has around 600 genes. If it had 10 genes, then it would be ranked as poor quality. If it had 10,000 genes, then a flag would come up to say it was poor quality. So if the N50 and the L50 were poor numbers too, those would also be reported. I hope that answers the question. Okay, so this is the information about the annotation. And then this is a standard thing that we do in PATRIC to let you know, and it's also something that you could put directly into your announcement for your genome. There were 28 hypothetical proteins and 577 proteins with functional assignments. It'll tell you how many of them have EC or enzyme commission numbers, how many of them have gene ontology assignments, the number of proteins that are in specific pathways, and we use KEGG pathways. And then here we have the genus-specific families and the cross-genus families. We call those PL FAMs and PG FAMs. Now, I'm going to try to shrink this thing down because this thing is, well, you know, it's hard to shrink down. But we provide you with a separate thing when this never works very well. So this is the Circos viewer of your genome. And so you'll notice that it shows the number of contigs here with the breaks here. This is the first contig. This is the second contig.
The outer ring.
The third, this outer ring. Third contig, fourth, fifth, sixth, seventh, eighth, 9, 10, all the way up to 17. These are the genes that are called on that contig. So the outermost ring, if we look at this, the outermost ring is the contigs. The second ring here is the genes on the forward strand. The third ring is the genes on the reverse strand. The fourth ring are the RNA genes. And I always use RNA genes because that's always an indication to me that tRNAs are associated with lateral transfer. And so if these are GC skew and GC content and GC skew, if you're looking for lateral transfer, if you can see where you've had a change in the GC content and you can see that that's tied to a tRNA gene, then I would suggest go look at that thing because it's probably lateral transfer. But anyhow, I get excited about that, so I digressed. The next one in are the known virulence factors. You'll notice in my genome, there are none. Well, this is an endosymbiont, and so its role is not to be virulent. Its role is to get along with its host. So it didn't have any virulence factors. And then it looks like one, and hits to known antimicrobial resistance genes. So forward strand, reverse strand, RNAs, virulence factors, AMR, GC content, GC skew. And so what is with these wacky colors? I don't understand what's going on with the colors. So you can go down here, and the colors are based on our subsystem categories. Subsystems are a group of, what we've gone in is we've identified genes that have shared functional characterizations. They're involved in the same pathway, or maybe they're transporters. So subsystems are a pretty valuable way of looking at data in PATRIC. It's something new that we've added to it. But you can see here that the blue indicate metabolism. If I go up, you can see where those genes are linked together. The orange are protein processing. You'll notice that in Buchnera, they do a lot of protein processing, and those genes are generally grouped together. Bacteria are just like, they make decisions based on energy and spending energy. And generally, if they have things that are doing the same functionality, they group them all together so that they don't have to waste energy with transcribing genes. So you can often see that a bunch of things are grouped together, like these green here are within energy, and those are grouped together on the forward and reverse strand. And here's a big group of metabolism genes. Specialty genes, we've talked a little bit about this. Every genome that goes through PATRIC, every genome that's annotated, and also every genome that we bring in from the public resource, they are blasted against specialty gene databases. We have them for antibiotic resistance. The source of that we use is CARD. We also have a special PATRIC gene database that we use. Drug targets. So if your particular, this is often of interest to people if they want to know if anybody has attempted to use a particular protein as a drug target. We blast against that database to provide you with that information. The transporter database, we take that from TCVB, and then we use three virulence factor databases. The third one isn't listed here because it didn't have any hits against it, but it's the FDB, PATRIC virulence factor database that we hand curated, and one called Victors that's another public resource. So the next thing we do, and the final thing that we do in the report, is we build a quick tree. What we do is we use the similar genome finder, which does MASH min-hash, to try to find the closest relatives to your genes. And then we choose 10 close relatives and we try to step out one. So you see, try to find the nearest one. And this is a very fast approach for a tree. I wouldn't use this necessarily in a publication, although I like this. It takes 5 genes, the protein, let's say 5 proteins that are single copy in each of these genes using the PATRIC global families. So it finds 5 proteins that each of these genomes have that are single copy. And it brings in those 5 genes and bumps them all together into a single long string. It also gets the genes, not the protein sequences, but the gene sequences for each of those 5 genes, and there'll be slight differences, based on the 3rd codon. And it puts those in, and so it builds the tree based on that. And these are bootstrap numbers. We only do 5 because it's really fast. And we want to give you hints about where you should go next in your analysis. So that's the reason we do 5. But then what I would do is I'd go to our phylogenetic tree program that does hundreds of genes. It does a blast analysis to find 100 of the closest related genes. So that's what I would do if I were you, to look at that data. So and then at the very end, it shows you the publications that are linked to the different processes. So this one is the first that recognizes PATRIC. Please use that when you publish your data, if you did your analysis in PATRIC, because we all live based on this paper being referenced. If you did spades, it'll give you the reference for spades. This is the reference for RAS-TK, which is the annotation pipeline. If you wanted to include that you had used the EC numbers, where we get our EC numbers from are Brenda, the enzyme database. So you can show that. I just want to make it as easy as possible for you guys to publish this information without digging through things. Because I'm a biologist myself. I'm not a computer scientist, and I recognize how tough it is for people to find out what publication of spades, what part of the pipeline was used. It's all here for you to make it easier for you. Okay, so this is our HTML. I'm going to go back to PATRIC. So let's say I walked away from this for a while. So that's the report part. The CIRCOS, this is back to the jobs list. And so there's a number of steps here. We looked at the HTML. Remember, I looked at that big circle thing and I said, oh my goodness, that's a mess. What am I supposed to do with that? Because it's really big. This will give me this won't. I've got to download this. This is a download version of this. So I can download this, save file, and I can go into my download folder and open that up. There, now I have it. It's an SVG. It is publication quality, and you can drop that into your manuscript, okay? You could also get it as a scaled vector graph. The codon tree, the NUIC, I don't know if any of you have ever looked at those, but the NUIC file is the directions for building the tree. So let's say you really wanted to use that tree. We could look at it here. That's going. It's going to take a little while to load. So I'm actually going to go back to that. But what I can do, I don't necessarily like the way that PATRIC builds the tree. And remember that HTML report, the branch on the tree was bright red. Let's say I wanted to do something really quick, but I didn't want to pay for color. I would take the NUIC file and I would build a tree in Figtree, perhaps, or ITOL. Those are two good resources for you to make the tree, to build the tree. These are the instructions for building the trees, but then you can adapt how it looks. Figtree is capital F-I-G, capital T-R-E-E, it's one word. And ITOL is the interactive tree of life. So those are mainly the two most important things. Actually, I'll download this and we can look at the SVG for the tree too. So if you wanted to really use that tree, it would, oh no, I don't want to open in Adobe Illustrator. Uh-oh. I've launched something that I didn't want to launch, but that's where you could build the tree. I know, I don't want to see it. I'm going to close that. But if you have something that you can view scaled vector graphs, and that's the place to do it. So that's basically the service. One more thing I wanted to show you is that Genome is now incorporated into PATRIC. So I can go in, let's say I'm lost. I want to find my Buchnera genome, and I can always look here. And this is actually a little bit slow in PATRIC these days because now we're having to, PATRIC, when we first started PATRIC, we had 8 bacterial genomes. Now we have 180,000. So, and more, we get 10,000 or more every month. So it's going up, up, up. So you can look for it this way. I often use the global search to find it. And then what I would do is I'd go in and look at the genome table, and then I could find all my private data. And that's what I'm going to show you how to do. So it's returned just going up here for Buchnera. You can see that there are 83 genomes, and it will show you 3 of them, the top 3. And if I wanted to see all of them, I could click here. Genomic features, these are all the genes across all those 83 genomes. But where I generally like to go is I would go to Buchnera here because I like to see things at the taxa. So I click on that. And then it will give me a summary of all the genomes that are in the database, including the reference and representative genomes. It'll tell me different hosts that they have been isolated from, the countries, and the genome status. But let's go in and look at the genomes, because let's pretend I can't remember what this thing is named. And you'll notice here, there are a lot of things with really suspicious names, but as I go down, I can see that there are a lot of plasmid genomes, whole genome shotgun, complete. If I click over here on the filters, I can quickly, under public, it looks like true, there are 59 genomes that are in the public database. If I click on the false, I see that there are 18 genomes, and false means it's private. That's computer speak for private. So now I know that I've annotated this genome 18 times, and so this would be the place to look for that private genome. If you can remember what you called it, you could also put it up here and search for it here in the view, in the global search. But once it's in here, I can use any tool in PATRIC. So I could go into, let me put in true here so I have all of them. I could use this tab for the protein families, and it'll summarize all that data across both my private and public genomes. So it'll all be summarized here. So once you annotate something in PATRIC, all that data is available for any analysis. You've got it all here to look at. You can look at the pathways. You can look at the subsystems. It's all there. So that's the benefit in PATRIC. So the new service will give you an indication of the quality of your genome, how good it is, and then give you a shot of, we think that here are the closest relatives. You might want to build a better tree with these. Here are the genes that appear to be functioning if you want to go look at pathways for different functionalities. Here are these genes you might want to pay attention to. But then if you want to do a true comparative genomic analysis, this is the place to do it. And once you've done it either at the comprehensive genome analysis, or if you've done it in the standard assembly and annotation, that data is in PATRIC and you can compare it to any of the 180,000 genomes that we currently have. So if you guys have any questions, I'm happy.
Did you want to show that file to the tree?
Oh, to get it to load?
The one that you, no, not the one you had to show in PATRIC.
Which one? Say it again. Sorry.
The one you had in PATRIC.
Ah, yes. Oh, there's a question of looking at one of the phylogenetic trees that was in PATRIC. Yes. Let me.
So you started that.
Yeah, I started that. Let me go back and filter on that. And I'll look at this one and I'll view it. I'm just stepping you guys through it again so that you can see it. And the Newick file, and I'm hoping it will view. So it's going to take a while to do it. But while it's doing that, I wanted to show you guys this thing that I really like, applications. And it's Fig Tree. This is the one I use all the time. So I could download the instructions for that tree. Let me see if I have one that's I opened recently. While I'm waiting for this guy, I'm going to go back really quickly. Do the Newick, download it, save it in downloads, which I hope it does. And then I can go into Fig Tree and I can say open. And let me look in, I think it was in downloads. Oh, that's Zika virus. I don't want to do Zika virus. I don't want to do that. Here we go. and we run the bootstrap. So, oh, sorry about that, everybody. I'm gonna get it for you here. So, an unimpressive tree at this point, but I can go in. This is what I like about Fig Tree. I can increase the font on it, so I can make it as big as I want. The bootstrap values are in node labels. I just called it support for here. So then I can see how well supported the tree is. And then I can get that as big as I can get that as big as I want. So Fig Tree is pretty awesome because you can go in and do a lot of things with it. And then like if I wanted to root the tree on this guy, which is here, I can root that, and then I can rotate. So I can make the tree look exactly the way I want it to using Fig Tree. If I wanted to do midpoint rooting, I could go into Tree and do midpoint root, which is what that's what's happening in PATRIC in the HTML that we saw. So there it all is. That's my favorite one. A lot of people like interactive tree of life because you can have metadata along the sides, but Fig Tree is easiest for me to use, so I haven't really worked that hard at interactive tree of